Porifera, as well as the general features and classification of Porifera. So, starting with the Porifera, if you go through the previous year questions, you will notice that the questions have been asked specifically from skeletal system, for example, skeleton in sponges, general characters and classification of Porifera. This question has also been asked. So yes, even if this topic is not mentioned in the syllabus, you have to prepare it because it has been asked, and it's a general uh, way to prepare for the invertebrates and vertebrates. The general way is that you have to know the classification. This is mentioned in the syllabus classification, so you need to know the classification at least up to the class level in all the phyla. And then yes, the things that are mentioned in the syllabus. So yes, there are questions regarding the skeletal system. Here again we have the question based on spicules, that is part of the skeleton system. And then you can see the questions related to the canal system. We will be discussing it in detail, the canal system, the skeletal system and the reproduction. So in this lecture we will be discussing about the general feature, the skeletal system in Porifera, the canal system and the reproduction. So starting with the Porifera, Porifera, these are also called as the sponges. And porifera, the word pore, it means pore, pore bearing. These are the pore bearing organisms and they contain pores in the body. If you will see the structure of this porifera closely, they have small pores on their body, and these pores they are called as ostia. And you will observe that these are cylindrical in shape. So these are cylindrical in shape and these are pore bearing. These are also the first multicellular animals. After the protozoa, we have to study the porifera. So these porifera, these are the pore bearing animals. In porifera, in the phylum Porifera, we have different classes, and these classes, the classification, it's based on the spicules. So on the basis of spicules, the phylum Porifera, it has been divided into three classes. After the classes we will discuss the general features. We can do it first and then the classification, but it's okay, let me explain it here only. So we have three classes. The three classes contain class Calcarea. Calcarea, this word Calcarea, it is related to calcium carbonate. So the skeletal system in the class Calcarea, it's made up of calcareous spicules. These are radially symmetrical. I have explained you that what is radially symmetrical. Radially symmetrical, they have a central axis and the body can be divided into equal halves around the central axis when the plane passes through the central axis. So these are radially symmetrical. Then they contain conocytes. The conocytes are prominent and easily visible in case of the Porifera. The body has single spongocele and opening via the osculum. We will be seeing the structure of the Porifera. We will be discussing it here, that this osculum is the uppermost, it is the opening from where the water comes in, from where the water goes out, osculum, because ostia are the holes from where the water comes in. So I'll explain that, let it be for now. But the thing is that in Calcarea, body has a single spongocele opening via an osculum, and you will find it in all the sponges that there is spongocele. And example of Calcarea is Sycon, Leucosolinia and Clathrina. Then we have class Hexactinellida. In Hexactinellida, the skeleton, it forms 6-ray triaxon siliceous spicules. Okay. The canal system, it may be branched or unbranched. And the organism, they show radial symmetry. It's commonly called as the glass sponges, for example, Euplectella. Then in case of Desmospongia, this is the third class. This class contains the spongin fibers or the siliceous spicules. So Desmospongia contain the spongin fibers or the siliceous spicules, and they possess the raygon type of canal system. For example, Spongilla and Cliona. So these are the three classes of the Porifera that you need to know. Then let's discuss the features. First of all, you look at the diagram of the Porifera. You will see that there is this one opening that is called as osculum. Okay. Then there is this outer layer. This outer covering, outer layer, it's called as a pinacoderm. Okay, it's called as a pinacoderm. After the pinacoderm, you will observe these small holes on the body. These holes are called as the ostia. Ostia are the holes from where the water enters into the body of the Porifera. Inside, you will observe this cavity. This cavity is called as spongocele. So this cavity is the spongocele. This cavity is spongocele. Inside the spongocele is lined by the layer that is called as the conoderm. Conoderm is the layer because it consists of the conocytes. These conocytes, these are the conocytes. These are flagellated cells that are lining the spongocele. So these are the flagellated collar cells. Inside, between the pinacoderm and the conoderm, there is a central, the middle layer that is the, not the central, but the middle layer that is the mesohyl. It is the mesohyl is the layer between pinacoderm and the conoderm. So these ostia, they go inside the spongocele through the porocytes. There are cells that form the pores that surround the pores. They are called as porocytes. There are certain cells which are which have the potential of totipotency, which can develop into different types of cells. These are called as the amoebocytes. Then they also contain the spicules, different type of spicules or skeleton inside the body. So these are different types of cells and different types of layers present in the Porifera. So this is the general structure of Porifera. The general features of phylum Porifera, these are multicellular, but they lack a true tissue or organ system. As we saw in the diagram, there was not any true tissue or organs present. This body may be asymmetrical or radially symmetrical, and its body is cylinder-like. The body is asymmetrical or radially symmetrical. The pores, that is ostia, and there is single exit pore. So ostia was the pore from where so ostia is the from where the water is in current, goes inside the body, and the exit pore is the osculum. Then these are mostly sessile. Okay, they are fixed at one place. These are mostly marine, and some of them are fresh water. Then the body is made up of two layers that I said earlier. One is pinacoderm, other one is inner conoderm, and this conoderm, it is lined by the conocytes. Skeleton has spicules in it, calcareous and siliceous spicules, and contain the spongin fibers. Then the canal system, it's present for water circulation, and it has three types of canal system: Asconoid, Syconoid, Leuconoid. We have to do it in detail. They have collar cells or the conocytes, and these conocytes or the collar cells, they line the inner chambers and help in filter feeding. And then the digestion, it's intracellular. It has intracellular, it happens within the cell. And there is no circulatory system, no respiratory system, no excretory system. The asexual reproduction is by budding or fragmentation. We will have to discuss it in detail, the reproduction type, the asexual as well as sexual. The sexual reproduction is hermaphroditic, and the fertilization, it's internal, because this organism, it contains both male and female. Same Porifera, same sponge will be able to produce both ovum as well as sperm. So development includes a free swimming larval stage, that is amphiblastula or the parenchyma, and it has high regenerative capacity. So these are known as the pore bearers due to the pore exam, that is the skeletal system. As I have already told that many questions have already come from this topic, so we will, and you will see that almost every year some questions have been asked in for forest services or the CSE. So the topic is skeletal system, skeleton in the Porifera. The skeleton of the Porifera, it is secreted by special cells, and these cells are called as the sclerocytes or the spongocytes. Okay, and these cells originate from the archaeocytes. Archaeocytes are the totipotent cells in the mesohyl. So here I would like you to make one correction here, that I said that amoebocyte, but there are archaeocytes which are the totipotent cells in mesohyl. So mesohyl is the middle layer between the conoderm and the pinacoderm. The types of skeleton on the basis of the composition, on the basis of chemical composition, the skeletal system is of three types. First one is calcareous, that is involving calcium carbonate. It is found in class Calcarea. And spicules, they can be monoaxon, like they have one axis, triradiate, tetraxon, that is four rad. We will study about them. And the examples for calcareous skeleton is Sycon and Leucosolenia. Then for siliceous skeleton. Siliceous skeleton, they are composed of silicon dioxide, and they are found in hexactinellida, they are found in desmospongia. Hexactinellida and demospongia. So the hexactinellida spicules, they are six rayed. Hexa, the word hexa, it denotes six. Hexa means six. So these are six rayed. And hexaactine, and they are often fused into lattice structure that is found in Euplectilla. If you will Google the diagram of the Euplectilla, you will find it has this structure that is made up of spicules, siliceous spicules. Then we have demospongia. These have non-six rayed and they do not fuse. They are not fused. For example, in case of spongilla. Then next we have spongin skeleton. In case of spongin, there is a flexible collagen-like protein, that is the spongin itself. Spongin is a collagen-like protein, and they are found mainly in demospongia. They provide elasticity as well as softness. At the same time, we need to know that yes, these skeletal system, they provide the strength, support to the organism as whole. Spongin skeleton is found in Euspongia, that is called as a commercial bath sponge. Next is mixed skeleton. It is found in Demospongia. They have spongin fibers and siliceous spicules. It offers both rigidity and flexibility. Now discussing about the types of the skeletal system, types of these components. So the spicules, they are generally of two types. One is the megascleres. Second one is the micro. Mega are larger in size and micro are smaller in size. So when we discuss about the megascleres, these are basically the larger ones. These are larger skeletal spicules. They constitute the framework. They form the supporting framework of the sponge. Now these are generally of 5 types. 5 types. The 5 types are monoaxon, tetraxons, triaxons, polyaxons, and spheres. If we see the monoaxons, you will observe that the growth of this spicule is along a single axis. Monoaxon can be straight as well as curved. So the monoaxon, they can be like this. This is diactinal monoaxon. This is monoactinal monoaxon. This one is curved monoaxon. So these are the types of monoaxons present. Then after the monoaxon, we have second type, and the second type will be tetraxons. The tetraxons, they are also called as the tetraactines. These are quadriradiate. They have 4 rays. Like in monoaxon we had 1 ray, but in case of tetraxon we have 4 rays. So these are quadriradiate. And generally what happens, generally there is this 1 ray which contains a crown-like structure. This crown-like structure, it's called as a rhabdome. The crown-like structure, it is called as the amphidisc. And there is this rhabdome, that is the extended ray. Will be clear in the diagram. This is the tetraxon. This is amphidisc. After the tetraxon, the third type is the triaxons. These are also called as hexagonal spicule. So it has 3 axes and 6 rays. So it will be somewhat like this. So they contain 6 rays. These are the triaxons. And yes, there are certainly some polyaxons. They have a central point and many rays arise from this central point. Then we have spheres. These spheres, these are rounded bodies. And they have